Now connected from the phone. Can you hear me?
Very, very well. Thank you.
It's
Thank
me
you
too
very
now.
much.
Good.
Yes.
Very,
Okay.
very well. Very, very well. Thank you, Rashida. Thank you for the suggestion. It worked.
It's a good suggestion, Rashida. Okay, so you know that this presentation is about life science. From its title, it says that from DNA to protein, and this presentation will be presented by me for the first part, in which I will talk about transcription, which is a step of this dogma, which is going from DNA to protein to produce the protein. The second part will be presented by Mohammed Al-Jazari. He will talk about translation, second part. And the third one will be presented by Mohammed Al-Hayani. He will talk about a technique which is used in molecular biology related to DNA. And finally, Mr. Mohammed bin Qaid will test you, will assess you if you understood anything about what have been already said. Okay. So let's begin my presentation. We can call it my lesson. The first part is about transcription. So before beginning this part, I have to ensure that you have some basic information about this topic, which is first, I can give you this question. What is a cell? Hello, Abdelhadi, welcome. Can you guess the definition of cell? You can write it in the public chat, or you can talk loudly. We can see you, we can hear you. What is a cell?
I think a cell is the smallest unit in the body.
In the body, smallest unit. Okay.
Yeah.
What else?
It contains all informations.
What?
It contains all informations in the what she's called in. She's called what she is in the middle.
In the nucleus.
Yes.
Okay, good. Yusuf said the smallest structural and functional unit of an organism, typically microscopic and consisting of cytoplasm and nucleus enclosed in membrane. Yes. Microscopic organism typically consist of a single cell, which is either eukaryotic or prokaryotic. An enclosed cavity in an organism, a small compartment in a larger structure such as a honeycomb. So you're a biologist, Yusuf. Are you cheating? Plagiarism, yes. That's it. So another smallest alive thing in our nature. So it characterizes the living the living being. Okay, so cells are the basic building blocks of all living things. The human body is composed of trillions of cells. They provide structure for the body. Okay, so nutrients from food, convert those nutrients into energy and carry out specialized functions. Cells have many parts, each with a different function. Thank you, Rashida. Main component of every kind of life. Okay. Yes, the basic membrane bound unit that contains fundamental molecules of life and which all live. Okay, good. So right, so the cell is the basic unit of life, which is the origin of living being is, as you know, is a cell, which is this cell. It is called zygote, which is a fertilized egg cell. It's a combination of the sperm cell, which comes from the male, and the egg cell. It is called also ovum, which is the gamete. The gamete comes from the female. So the origin of life is a cell, and this cell undergo many divisions. As you can see here, the first division give us 2 cells. The second division gives us 4 cells, and so on until given, which is called the embryo. So the embryo, and after the birth, it is called the baby. So which is it is called this one. It is called fetus. These steps are called embryo, fetus, and after birth it is called a baby. So you can notice that living being, a human being, for example, mitosis, yes, division, it equals mitosis. So you can notice that the human being, for example, or in general living being, it is originated from only one cell, and this cell is divided, undergoes mitosis, divisions, many divisions, until becoming a multicellular organisms. Okay, so another thing, we can see here this schema, which shows us a cell, the structure of cell, a simplified structure of cells. So it contains a cell membrane, the cytoplasm, and the nucleus. And this nucleus is containing, which are called chromosomes. So the chromosomes are considered the support of the genetic information. So support, it means it carries all the necessary information to produce proteins. The proteins which are responsible or control the proteins control the traits. Okay. So I said that each chromosome traits features. Yes, Yusuf. Each chromosome, it is composed or made up of a long strand of DNA. So each chromosome, it contains two strands of DNA. This DNA is called enroulé, coiled, and condensed around nuclear proteins, which are called histones. They are spherical proteins that you can see in here. Okay. This long strand of DNA contains many genes. So the DNA contains many genes. Each gene carries the instructions to produce a protein. I'll ask you this question to ensure that you understood what I have said. Where are the genes located in the cell in general? Can you answer? Badruddin, yes, the male spermatozoon who determines the gender of the baby. Yes. So we have two kind of spermatozoon of sperms provided from the male. So the one who provide the XY chromosomes. So the sperm which have the Y chromosome and the other which have the X chromosome. So, okay, so the question was, where are the genes located in the cell? The core, in here, the core, do you mean, Marwan, the nucleus? Right?
Yes.
Yes, okay, so the nucleus. Yes, it is in the nucleus. Okay. And where exactly in the nucleus? Which is the support? No problem, Anas. Genes are located on the chromosomes. Excellent, yes. It is located on the chromosomes. And each chromosome contains a lot of genes. And each gene is responsible in the chromosomes. Okay, so each gene contains all the information, necessary information to produce a protein. Okay, so where does the synthesis of protein take place? Can you guess? If the gene is responsible for the synthesis of a protein. Synthesis, it means the production of a protein. Can you guess where can be take place? Is it in the nucleus, or in the cytoplasm? You know the gene, it is located in the nucleus. So the proteins, where can be produced?
Cytoplasm.
Cytoplasm. Okay, so we can repeat the question, Marwan. Okay, nucleus. Nu'man said the nucleus. It's logical because the gene, it is located in the nucleus and must be produced in the nucleus, but it is not the right answer. So, so we'll find the answer to this last question in the next slide. This is another figure that we can read. Who can read this text to answer the last question? Yusuf, during the mitosis. Why? Okay, Norman, we will figure out this. During the mitosis, what do you mean, Yusuf, by this? During the mitosis?
Maybe the production of proteins during the division.
So it's not during the mitosis. No, it's not during the division. It's before the division.
Okay.
Okay. So who can read this text?
Can I read, teacher?
Yes, Rashida, go ahead.
Studies have shown that protein synthesis takes place in this cytoplasm through the invention of cellular structures called ribosomes, while the genes are that control protein synthesis are located in the cell nucleus. Some experiments have shown that DNA does not come out of the nucleus, so protein synthesis requires sending information from the nucleus to the cytoplasm. In order to verify the existence of such message, Goldstein and Plot, 1955, carried out the experiment shown in the figure opposite. The role of the radioactive 22P is to label the DNA.
Okay, thank you, Rashida.
Welcome.
Okay, so so the answer of the last question is the genes that control protein synthesis are located in the cell nucleus, and studies have shown that protein synthesis takes place in the cytoplasm. So this is why. So experiment studies showed that the protein synthesis, it is Norman, it is produced. The proteins are produced in the cytoplasm. So we know that the gene, it is in the nucleus, and the proteins are produced in the cytoplasm. The question now is which is what is this molecule intermediate between the nucleus and the cytoplasm? Why? Because we know that DNA, as an information that you have in this text, the DNA does not come out of the nucleus. It means that cannot leave the nucleus during the synthesis of the protein. So we have to guess the nature of a molecule that plays the role of an intermediate between the nucleus and the cytoplasm. So we have many information to guess what is the nature of this molecule. We can look this figure that shows us the experience of the experiment, that is led by Goldstein and Plaut, 1955. So the first thing is the amoeba A1 in a medium containing radioactive phosphorus 32. It's important to notice that the P32 or the phosphorus enter into the composition of the DNA and RNA. The phosphorus enters into the composition of nucleic acids. So they are called nucleic acids because they are found. They were found in the nucleus. So the amoeba A1, it is called amoeba A1. It is in a medium containing radioactive phosphorus 32. We use radioactive phosphorus because it allows us to label the DNA, to know where this DNA or where this, to label the acid nucleic, trace, yes, trace, to know where is this nucleic acid goes, the pattern of the acid nucleic. It could be the DNA or the RNA. So we made this amoeba A1 in a medium containing radioactive phosphorus 32. After a while, we notice the appearance of radioactivity in the cytoplasm and in the nucleus. So what does it mean? If we make an amoeba in a medium containing phosphorus 32, in a while it enters through the membrane. Yes, excellent, Yusuf. That means that the phosphorus 32 passes through the membrane into the cytoplasm, infiltrate. Yes, and after that, where it went, or they went, if we talk about phosphorus molecules, into the nucleus. Yes, good. Another step in this experiment, we have an enucleated amoeba, A2. Enucleated, it means we use an amoeba without a nucleus. We remove the nucleus. We remove the nucleus of another amoeba, which is A2, and after that, without core, without nucleus, yes. So we transfer the nucleus of the amoeba A1 to the cell, the amoeba A2, which is enucleated. And after a while, we notice the appearance of radioactivity in the cytoplasm of A2. So what can we infer? It means that there is a communication. The amoeba can travel through the cell from nucleus to the outer space of the cell and back. Yusuf, here we are talking about the phosphorus 32, not the amoeba. The amoeba can go into the nucleus and then come back to the cytoplasm, right? This is what do you mean? Okay, so good. So there is a hypothesis. We have to make a hypothesis concerning the nature of this molecule. What is the nature of this molecule? So the first question, knowing that there is another information, knowing that phosphorus enters into the composition of nucleic acids, DNA or RNA, and by analyzing the results obtained, propose a hypothesis on the nature of the message transmitted between the nucleus and the cytoplasm. So why the phosphorus is getting out? Okay, so there is a message. There is a message that go from the nucleus to the cytoplasm. And this message must be one of the two things, of the two molecules. We know that the phosphorus enters into the composition of nucleic acids, DNA and RNA. So here we have to guess which is the nature of this molecule. Is it DNA or RNA? Because the presence of phosphorus, radioactive phosphorus 32, it means that the nature of this molecule is an acid, is a nucleic acid, but we don't know if is it DNA or RNA. So because look this. This molecule, there is a trace of RNA outside of the nucleus. What do you mean, Marwan? Outside the nucleus. Why RNA? Because the DNA can okay, very good. This is the second. This is the second information that we have to take into consideration in order to propose a good hypothesis. The DNA does not come out of the nucleus, and the second thing is that the composition of phosphorus enters into the composition of nucleic acids, DNA and RNA. So the only molecule that can be in this condition, in this situation, the only molecule that could be the intermediate between the nucleus and the cytoplasm is RNA. Okay, is it clear? Do you have any question?
Not for me, it's great.
Abdelhaq, I didn't. Sorry, but I didn't get the last idea.
Okay, the idea about the hypothesis, the nature of the message transmitted between the nucleus and cytoplasm. Okay, yes. We have in this experiment and the text, we have two necessary information to understand the nature of this message. What do I mean by the message? There is a molecule that must go out of the nucleus and go into the cytoplasm in order to produce the protein. we said that the gene, it is in the nucleus and cannot get out of the nucleus. So it must be there a molecule intermediate that come from the nucleus going to the cytoplasm. And the type or the nature of the nucleus, the big density of DNA is higher. Okay, I continue. So I said that the nature of this molecule can be guessed using these two informations. We know that the radioactivity that appears another time in the cytoplasm must be of two things, one of two things, which is DNA or RNA. Why? Because the composition, this phosphorus enters into the composition of nucleic acids that you have in the first question in here. So it could be DNA or RNA. Why we have to say it is an RNA? These messages, these molecules are RNA and not DNA, because we already know that DNA cannot leave the nucleus, cannot come out of the nucleus. Is it clear now?
Yes. Thank you so much.
Okay, so this is a good information to understand the nature of this molecule. And the second question, by treating, we have to confirm this question, this hypothesis. By treating the cell with RNA, degradation enzymes, we observe the arrest of protein synthesis. These are further information by which we can confirm if the hypothesis is true. We treat the DNA is the post office and RNA is the postman. Yes, exactly. So, but can you answer this question? Does this experiment confirm the previous hypothesis? What do you think?
Yes, because there is the protein, the rest of the proteins.
The arrest, yes.
Yes.
There is no production of proteins because of the use of RNA degradation enzymes. What is the role of these enzymes? The degradation enzymes break down the RNA in the cell. So if we noticed that if the RNA in the cell are broken down, so we observe the arrest of protein synthesis. We destroy the RNA. Yes, exactly, Abdelaziz. If we destroy the RNA, the result is the arrest of protein synthesis, of protein production. So, right. So this experiment confirms the previous hypothesis, because the arrest of protein synthesis indicates that the degraded or the destroyed substance is the one which is responsible of the production of proteins in the cytoplasm. Okay, so now let's move to know what is the difference between DNA and RNA. No RNA, no protein. Excellent, yes. So we have to know in this slide what is the difference, or what are the differences between DNA and RNA, and also similarities if there are some similarities. So can you tell me what are the differences, or what the differences and similarities between the two molecules are? Based on this figure, or the two figures, RNA is single, right? exactly. And you can use the expressions that we have learned last time. Okay, so RNA is single, DNA is double. No man, yes.
There is
Rashid?
different component in the DNA nucleotide.
Nucleotides, yes.
Ribose and deoxyribose.
Okay, so the nucleotide in the RNA, it contains a sugar which is named ribose, right?
Yes.
And in the DNA nucleotide, we have a sugar which is called deoxyribose. Okay. Rashid said AGCT in for the DNA. Okay, and the other is AGC N. Do you mean you? RNA contain OH. Yes, Abdulaziz. This is the difference why it is called deoxyribose, and the other it is called ribose, because the sugar in this DNA, this sugar doesn't contain the oxygen, the atom of oxygen. Because of that, it is called deoxyribose or deoxyribose. Abdulaziz, RNA contain OH, yes. Yusuf, while, whereas. Okay, so and DNA contain H only. Yes, Abdulaziz, in that point, similarly alike. DNA is a double-stranded molecule, while RNA is single-stranded molecule. Yes, exactly, Marwan. Nitrogenous base are different. Nitrogenous base are different. Are all of them different in the two molecules, DNA and RNA? More components than RNA. Okay, so it is single-stranded for the RNA, and the DNA is double-stranded. Yes, Rashid, it's you. The uracil is the different nucleotide, which is specific for the RNA. Yeah, and the one which is specific for DNA is thymine. Yes, thymine. Okay, so RNA for uracil and DNA thymine. Yes, Marwan. Okay, good. You are good students. Excellent. So the similarities. Is there any similarities, or there are any similarities? A, C, and G. Okay. And all? No. What does this mean, Yusuf? In A? None. Okay. So the only similarity is that the two molecules contain nucleic acids. Nucleotides. So even if there is a difference in, for example, the uracil, which is specific for RNA, and the thymine is specific for DNA, but the two contains the nucleotides. So we can see that the two molecules consist of nucleic acids. Okay. As you said, for the differences, DNA is double-stranded molecule, while the RNA is single-stranded molecule. The nucleotides in DNA contain deoxyribose, while in the RNA contain the ribose, which is sugar. The thymine is specific for DNA, and the uracil is specific for RNA. Unlike all types of RNA, DNA has something, if we are talking about the molecular weight, the DNA has high molecular weight or molecular mass for the teachers of physics. So why it is logical? Because the DNA is made up of 2 polynucleotide chains, 2 strands. Okay. So the composition of DNA. Let's focus on this a little bit. Molecular mass. Yes, molecular. We can see also molecular weight or molecular mass. Okay, so I said let's focus on the composition of DNA. So the DNA is, as we said before, it is double-stranded. It means that it has 2 strands, 2 helix, so double helix. It is composed of nucleotides, each of which contains a phosphate group. This one, phosphate group, and a sugar molecule, and nitrogenous base, And based on the type of nitrogenous base, we can obtain 4 types of nucleotides in DNA. So we can obtain nucleotide with adenine, nucleotide with guanine, nucleotide with cytosine, and nucleotide with thymine. Adenine is in this DNA, in this double-stranded structure. Adenine always bonds with thymine. So adenine in the first strand is or bonds with thymine in the second strand, and the cytosine is always bonds with guanine. So cytosine bonds always with guanine. While the composition of RNA, I said that it is single-stranded molecule. Also, it is composed of nucleotides. So they are similar to the nucleotides found in the DNA, which are adenine, guanine, and cytosine. And this nucleotide, which is uracil, replaces the thymine in the DNA, and which cannot be found in the this thymine cannot be found in the RNA, and the uracil also cannot be found in the DNA. So let's move on now to our real topic, which is transcription. We have to read first this text before answering these 2 questions. Can someone read this text? Norman is remembering high school in English. Okay. Yes, A with T, C with G. Okay. Marwan is raising his hand. I cannot see. Marwan, read, please. I cannot see the raised hand.
Okay. Miller's trees have been seen using the electron microscope. The Miller trees are produced by transcription of the genes. The trunk of the tree is made up of DNA, and the branches are the different RNA molecule being synthesized. These are shorter towards the start of the transcripted region and longer towards the end.
Okay, so the question based on figures 2 and 3, 2 and 3, this one and this one, determine the stages of transcription. The first question before, so we can divide this question into many questions, many small questions or short questions. First of all, by observing these figures, what is a transcription? What do you think is transcription? Capian? Capian what? Exact production of RNA from DNA. Exactly, Abdelaziz. Yes. Translating it is another word that we will understand or we'll see with Jazari, with Mr. Jazari, from one language into another. So it's a way of translating. Yes, exactly, Yusuf. But the term translation, it is used in the production of proteins, not in the production of RNA. Okay. So the transcription, okay. The transcription, it is so a mechanism allowing the synthesis of what? RNA. The synthesis of RNA. Because this RNA will be like reading a message. Yes, exactly, Marwan. This molecule is a messenger. Is a messenger because it is sent by the DNA, the gene in the nucleus, to the cytoplasm to be read. So because of that, we call it RNA messenger or messenger RNA. Okay, messenger RNA. So this messenger RNA molecule, it is produced from the gene. And the production or this transcription, it takes place in the nucleus. Okay, yes, mRNA, Yusuf. mRNA, messenger RNA. Okay, good. Another question. We said that this happens, where does the transcription happen? We said that it happens in the nucleus, right?
Yes.
Okay, so we know that we have 2 strands in the DNA. Okay, so 2 strands. Which strand is susceptible? We can see this susceptible of being transcribed. Susceptible of being transcribed. From which strand the mRNA is produced? So from this figure, you can infer the answer. Template strand. Exactly, Marwan. It is called also transcribed DNA strand. It is called transcribed DNA strand or template strand. And in the opposite, there is the other strand, the transcribed DNA strand. This one, it is oriented 3' end, 5' end. Okay. What is Abdelaziz, subhanallah? How to distinguish? So yes, how to distinguish the 2 strands? Yes, okay. So this strand, which is called transcribed DNA strand, we cannot determine it. It's not we are us or who can determine which strand can be, which is the transcribed DNA strand or the template strand. It is known or recognized. It is recognized by this molecule, which is RNA polymerase. If we want to produce a machine that do the same, how it will cost? Yes, it depends on the RNA. So we can. Okay, good. So we said that the RNA polymerase recognize a specific sequence of nucleotides in the mRNA, in the DNA, in the transcribed DNA. So RNA polymerase, it's responsible for recognizing the right strand that can transcribe. Okay. So we have here two strands: the transcribed one and the non-transcribed one. So what else? So another question. We said that mRNA is synthesized from one of the two strands that make up the DNA double helix. So the strand that serves as a template is called a transcribed strand. Yes. So we can produce it. What is the enzyme from the figure? Exactly, figure 3. What is the enzyme responsible for doing transcription? Can you answer this question? What is called this enzyme? Enzymes are proteins. They have a function to lead. So the function of this enzyme is to produce the RNA, the mRNA. What is called this enzyme? Check in the figure 3. RNA polymerase. Exactly, Marwan. Thank you. So the RNA polymerase is the molecule responsible for producing the mRNA. And in which orientation? Is it in 5', 3', or 3', 5'? 3', 5'. This is the orientation of the transcribed DNA, the template strand, 3', 5'. Okay? But the transcription goes from 5' to 3'. Okay, Marwan. 5', 3'. And remember this information, it will be in the quiz. I'm cheating. Okay. You're welcome, Norman. Okay. From where this enzyme begins the transcription, it begins from so what is called the transcription initiation site. Initiation site, and it ends when it comes or arrives to the transcription stop sites. What is the next or the step? Next step. So it begins. The enzyme begins from the transcription initiation site. After that, what is the next step after recognizing the initiation site? Yes, Abdulaziz. Sorry, I took a lot of time. The elongation, yes, exactly, Badruddin. The elongation or the transcription, and which is the step. So the next step after the recognition of or after recognizing an ancient site is that the RNA polymerase separates the two strands of DNA. So by breaking the hydrogen bonds between the two strands. After that, it carries out the polymerization or the elongation, yes, of nucleotides by respecting, of course, the principle of complementarity of nitrogenous bases, which is U in front of A, U, which is the nucleotide specific for the RNA, comes in front of the A in the DNA, and A in front of T, G in front of C, and C in front of G, of course. The next step is that the polymerization continues until the end of the gene. So this until the arrival to the transcription stop site. Sorry, I have to open the door. Sorry for this incident. I said that polymerization continues until the end of the gene, until the arrival to the transcription stop site, where the RNA polymerase detaches from the transcribed strand, so releasing the mRNA molecule. Okay, so the mRNA passes into the cytoplasm. After that, it passes into the cytoplasm in order to be translated to proteins. We have this second question, which is shows figure 4 shows a fragment of an active gene. Give the nucleotide sequence of the mRNA resulting from the transcription of this fragment. So can you? Okay, so the answer, it is already here. The T or the A in the mRNA comes in front of T, U in front of A, and so on. So from this template strand, this fragment of the gene, we obtain this fragment of mRNA. Do you remember the complementarity from the nucleotides in the DNA and the nucleotides in the RNA? Can you remember? Okay, so good. And I'll pass the presentation to Mr. Mohammed Al-Jazari, and thank you for your kind attention.
Thank you